Hello, I'm Sarah, and this is lecture 2 in a series of 4 presentations about PSSM, which is polysaccharide storage myopathy. Solely talking about type 1 here. There are other types. So welcome. I hope you learned something and that it will be helpful to you and your horses. Okay, this is my horse. Isn't he beautiful? That's him at Loxwood Joust, having a good gallop. He's a purebred Appaloosa, and he was diagnosed with genetic disease PSSM type 1 in November 2015. So how does a horse get PSSM? The fact is that a horse doesn't get PSSM, as PSSM is a genetic disease. What we mean by this is that the horse is born with a faulty gene in every cell of its body, inherited from its parents. PSSM is passed from one or both of the parents who themselves have the faulty gene. They may or may not have symptoms, and the breeder may or may not be aware of any issues, but there is no other way a horse can be positive for PSSM1 except via the parents. So if neither the mare nor the stallion in a mating have the faulty PSSM gene, then the offspring can't be born with the disease. You can only be sure of this by genetic testing, which we will come on to shortly. There's a lot of argument about this issue amongst breeders who may have invested a lot of time and money into certain breeding stock, only to find that some buyers want a horse testing before they will buy and then pull out of the sale if the results come back positive. But as you'll be aware if you speak to enough people with PSSM positive horses, owning one or caring for one is not for the faint-hearted. Costs and disappointments can be very high. Some horses show very few symptoms, that's true, but others also are very disabled, and any PSSM positive horse can potentially become ill very quickly when their management is not ideal, and sometimes even when it is. Owners can find this distressing and upsetting when their beloved horse is so poorly, and it isn't something you'd want to volunteer for without full knowledge of what you're taking on. Now, I consider myself very lucky, so as you can see, my horse can perform, and he suffers much less than lots of PSSM1 horses. But I still wouldn't own another one with the same disease, especially since it's simple and accurate to test for now. Now, my breed of choice is the Appaloosa. I absolutely love them. I think they're a great breed. And crosses with quarter horses in Appaloosas are common. And it's in the quarter horse where the faulty gene seems to have originated. We're not quite sure of that yet. And the breed has a proportion of horses with PSSM. So Appaloosas with this breeding can get the gene from there, from the quarter horse inheritance. And draft horses as well have PSSM in their pedigrees, some draft horses. So if I was buying another Appaloosa or a quarter horse or a draft horse, I would personally always test before buying, and I would only buy a clear animal. Let's talk about that faulty gene then. So this faulty gene which causes all the trouble, what do we mean by this? We need some biology here again, but again, stick with it, as the more you understand, the better equipped you'll be to manage a horse with this condition. I get really excited about these kind of things, but bear with me because I love biology and I think it's totally miraculous. But yeah, it makes my brain hurt sometimes. Fair enough. So our genes contain the information our bodies need to control every part of our physical existence. Every reaction, every tiny bit of growth, every change is monitored and controlled, or put into action by our genes. So here's a picture of some chromosomes where our genes are located. You may have seen this from biology before. They're arranged in pairs. They're arranged in pairs, and in a horse there are 32 of them. Okay, so every cell, with a few exceptions, nearly every cell in your body and in any mammal's body really, there will be a fixed number of chromosomes. In the horse it's 32, and they're paired, so they have one that's very like them, and they stick together with them. And when it's dividing, they'll separate because half has to go into one cell, a sperm or an egg cell, and the other half goes into another, so that when they rejoin, you've still got the same number all the time, so that number stays fixed. But apart from that, this is how the chromosomes kind of thing will look if you wanted to examine them in a cell. So let's magnify this view of these chromosomes. Then we can I can demonstrate how the chromosomes put their instructions into action in the body. Now, chromosomes are tightly packed so that it can fit into the cells in our body because they contain hundreds and hundreds of different chemicals stitched together in a very specific way. But when they need to work, they need to unravel to expose the instructions for making all the chemicals that take part in the reactions in the body, such as producing insulin, getting rid of waste products in the kidney, and everything else that happens. So here's a picture of a chromosome unravelled. I'm ready to share its instructions with the cell it's in. Okay, so here at the top is the cell with the chromosomes in their pairs, similar to the last picture that we saw. And here's one specific chromosome, and it's very tightly packed. It's wound around different chemicals, basically to keep it safe and to ensure that it's not destroyed by any of the enzymes in the cell. And eventually, if you unravel and unravel and unravel, what you come to at the bottom is this ladder-like structure. Show you a bit closer there. This ladder-like structure. And that's where we need to focus our attention on next. So how it translates into a product, that is a protein or an enzyme or a tiny piece of bone or something like that, the building blocks are added together one by one in a very strict order. And the instructions are contained in that ladder. So for every 3 spikes in the ladder, that will translate into one part of a protein. So every 3, the instruction is add this amino acid to build up a protein. Then it goes on to the next 3 and it adds another one. So you can imagine with a large enzyme, which is maybe hundreds of thousands of those little building blocks put together, it can stretch out and it can be very long. So, for example, when we're looking at this, let's have a look at this picture again. So, for example, if we look at the cell needs to build a protein, so it needs to build, let's say, for instance, a piece of muscle, which is made of a high level of protein. It's going to need these instructions, which are in every cell. So, if we look again at the ladder bit at the bottom, this is this bit. This ladder will separate even further so that the instructions, the spikes, can actually be read, and sometimes they can be read both sides, which saves a bit of space. But what happens is every 3 prongs of the ladder, a certain very specific product is added to the chain, and then it moves on to the next one, and the next one, and the next one, and the next one, until it's actually creating something much bigger. So if we look at this protein, pretend we're making a bit of muscle. This is how it starts. First of all, it will start with a chain. So this is maybe this is the amino acids that form a chain to make part of a muscle cell. Maybe it's, what do we call it, myosin. This is how it happens. It starts off as a chain, and then then what happens is it folds in a specific way, because the reason why the amino acids, the building blocks that is, are very specific is because some like each other, some don't like each other. Some have a very specific electrical charge. They have an affinity for certain other chemicals which are not amino acids, such as sulfur or something like that. And this goes up to create this very specific shape. So the process goes along from here. So it starts off as a simple chain, and it starts to get folded, and it twists in a certain way, and parts move closer together and away from each other. And then we end up with something very complex. Which way are we? This way? No, this way. We end up with something very complex that actually is a piece of protein or a bit of a cell, or maybe an enzyme, which is actually a protein. And enzymes help different reactions to occur in the body. So, what could possibly go wrong? Since nearly every cell in the body has a complete set of exactly the same chromosomes, and there are literally billions of cells in a horse's body, or indeed in ours, many of which are replaced regularly. So think of how much skin your horse sheds. All that skin's got to be replaced, and it is continually. So all that reproduction of cells is going on. That's a lot of copying of chromosomes going on. And in the original cell which made up the horse, the sperm or the egg cell, sometimes a mistake is made in the copying of that ladder when it needs to be reproduced to be separated into another cell, because every cell has to have these chromosomes, and so they've got to be reproduced. The cell will check again that it's got everything right and in the right order before it will allow the cell to even think about dividing, otherwise it will reverse the process and start again. But a mistake is what has actually happened in our PSSM1 horses. So what has happened is one of those rungs in the ladder has been replaced by an incorrect one. So when it reads that instruction, it's placing the wrong building block into the product. So that protein that was twisting and folding, it doesn't quite twist and fold in exactly the same way. Now, this does happen all the time, and most of the time it's not even a problem at all. We have lots of mistakes in dividing cells. Usually they don't cause a problem, but in this case, in our PSSM horse's case, it has done. Because what it's done is, it's made the enzyme that is produced from this gene, which is called GYS1, that's the name of the gene, that's the list of instructions, located on a specific chromosome. The product that it makes isn't the same as a normal one. Now, it's only got one tiny change in it, in the string of thousands, but when it folds up, it doesn't fold up in the correct manner, and it makes the resulting enzyme slightly hyperactive, because two bits that shouldn't be quite so close together, they are closer together than they should be. So it means that the enzyme doesn't take a break. It doesn't need so much energy to make that reaction happen. So then we're on to our long chain of glycogen without the branching. That's what we're actually talking about. So what we call by that single change is a single point mutation, and that's the way that biologists will speak of it. One tiny change, and it's caused all these problems. It is totally unfair. So because of that GYS1 gene allowing this slightly faulty, slightly different enzyme to keep being reproduced in every cell, then we get our storage myopathy, so the glycogen isn't quite right. So if you want to go back to presentation 1, that is where we look at glycogen specifically and how that actually is made up and how it can't get broken down properly. So I'd refer you to that one if you want a bit more information about that, and I hope you enjoy that one as well. So the result of that is that because that enzyme is working too fast and the resulting glycogen in the muscles can't release the energy quickly enough when the horse is in hard work, the energy source for the cell dries up very quickly, and what our horses get is cramp. So horses are much more muscly than we are, but I don't know about you, I've had cramp and it is painful. Sometimes the effects can last 2 or 3 days. So you can imagine that the amount of muscle a horse has, every cell of its body has this myopathy. Every cell is producing this incorrect glycogen, and every muscle cell is short of oxygen when you ask them to work. So it's no wonder really that they get into problems, really, is it? So that's basically it for that little bit. I hope you enjoyed the biology. I can always answer questions if you want. Lecture 3 is about how we spot a PSSM1 horse. How do we know? What makes people think, I think my horse has an issue? So a bit more practical in the next one. Hope to see you there.